Go ahead.
All right, so I'm Tyler Bodily. I'm a third year bioengineering PhD, and the current interfaces trainee, and I'll be presenting on some work I've done in rapid diagnostics using ssDNA aptamers in conjunction with graphene field effect transistors for protein detection. My mentors are Gerd Kauenbergs and Ratnesh Lal. So my presentation will kind of follow this general format where I'll first discuss basic elements of electrochemical biosensors and specifically where graphene field effect transistors fit into this field. On top of that, I'll also introduce some data we've collected on SARS-CoV-2 protein and neurodegenerative protein detection using GFET biosensors. In addition, I'll also present some data on work we're doing to develop a GFET array interfaced with the CMOS integrated circuit for high throughput protein quantification. So the idea behind why biosensors are so critical for modern health is we have a number of different therapies and treatments we can do for an individual's personal health circumstances. And the only way for us to really narrow down what an individual person needs on a given basis is to figure out exactly what diagnostic kind of element they're being affected with. And the one way to do this is to look at different biomarkers they have in their system. You're all probably familiar with Fitbits and Apple Watches. They generally look at a lot of peripheral, external vital signs, those sort of biomarkers that can give some information about a person's health. But there are a lot of different biomarkers related to proteins within a person's body that aren't detected by external sensors. So that's where electrochemical biosensors can kind of fit that gap. So the first element of an electrochemical biosensor is the sample we're looking at. So typically with an electrochemical biosensor, you'll want to detect certain biomarkers. Those biomarkers can be related to proteins. So you can look for protein quantification. You can look for viral detection. You're probably familiar with RT-PCR and lateral flow, especially with the COVID tests during the COVID pandemic. You can also look at other non-proteins, non-viruses. Those are just by basic biomolecules related to maybe neurotransmitters or neuromodulators. So there's a plethora of different biomarkers that you can detect using electrochemical biosensors. So a lot of the work I've done has been in protein detection. Just kind of as an outline, some typical ways we'll look at protein quantification, protein detection is through ELISA experiments. Those can be quite tedious, where you have to basically adhere captured antibodies to the surface of a 96-well plate. Then you'll bind your target protein, your target molecule to those antibodies. Those antibodies will affix to those or capture those proteins. You can then use a secondary antibody for binding to that protein, which will have an enzyme or a fluorophore, some way you can basically detect a color change in the system or some sort of fluorescent signal that you can correlate with the protein concentration. But it can be quite tedious to get concentration results from ELISA, but that's a general and tried and true method for protein detection and quantification. There's gel electrophoresis. That's kind of another way, somewhat quantitative for measuring protein presence, and then other colorimetric tests as well, like BCA and Bradford assays. So for electrochemical biosensors, kind of like similar to an ELISA, we need some way to develop specificity and sensitivity to our target biomarker. And that's where this biomolecular probe comes into play. Typical biomolecular probes are antibodies, IgA, IgG, different types. You're all probably very familiar with that. They have different arms, 2 arms, sometimes multiple arms. These arms are specific to a target biomarker. They'll bind to them. And as the antibody binds to this biomarker, that can change the conductance of your biomolecular electrochemical biosensor. There are other probes as well, like enzymes are a good example. So you can have a target biomarker that when it interacts with an enzyme on the surface of our biosensor can produce some sort of, maybe through a redox reaction, some sort of outputs, some products, and you can measure the change in the concentrations of those products on the sensor's surface. There are other different probes like aptamers. These are DNA or RNA aptamers. They're a specific strand of DNA specific sequence of single strand DNA that folds into a particular conformation, and this conformation is what designs the specificity for a target biomarker. So kind of how ssDNA aptamers work, RNA aptamers are similar, but for single-strand DNA aptamers, you'll have a large library of different sequences of aptamers that you believe could be specific to your target molecule. You'll then develop those into aptamers physically. Then you'll mix those aptamers with your target molecule you're trying to detect or develop an aptamer that's specific to. Then these proteins, these cells, whatever you're trying to detect, you'll separate out the bound aptamer target biomarker, target protein complex. Then you'll basically separate out those aptamers and sequence them and update your sequence library, your oligonucleotide sequence library with changes specific to what aptamers sequences have the highest affinity to your target biomolecule. This will go on for many different rounds. Each round will produce higher and higher affinity aptamers. At the end, you should have a sequence for your single-strand DNA aptamer, ssDNA aptamer, that will fold into a particular conformation that when it interacts with a target molecule, will have a conformational change that will draw electrons to the surface of your biosensor and change the conductance, the conductive properties of your biosensor. So as I was kind of mentioning, we need some way to transfer that selectivity of our biomolecular probe, our biological element, to some sort of electrochemical signal. This can be done through FET devices, nanowire arrays, nanoparticles, etc. I'll focus mostly on field effect transistor devices. So field effect transistor devices come from the basic MOSFET design, where you'll have a source and a drain electrode, as well as a gate electrode. There'll be a channel that's an insulated channel that goes between the source and drain. And as you apply different voltages on the gate, you can essentially turn conductive properties across the source and drain channel on or off in a typical MOSFET design. Bio-FETs are a little unique in that regard, where you're not looking to turn the channel on or off, but you're looking to measure slight changes in conductance across this channel between source and drain in a fluid environment that is interacting with your biological sample elements. So one design for this is the graphene FET. Graphene has many properties that make it ideal for FET in biosensors. This is kind of a basic outline of what it would look like. So imagine this gold on the left would be the source, right would be the drain, and you can have a flow of electrons across this channel here, where the black part is the graphene that bridges this gap. Then you'd have a gate, sometimes to the side in plane with the electrodes of the source and drain, or sometimes it's on top in the fluid if it's a liquid gated configuration. Or it can also be, there can be an oxide layer, insulation layer between the two, where you can have the gate underneath your graphene channel. The idea being that there are many different configurations for where the gate and your graphene channel are in relation to each other. But the general idea is that this gate, you'll then modulate the gate voltage in such a manner that you can measure the change in conductance across this graphene channel. And as this graphene, the surface of this graphene is modulated by different biomolecules binding and affixing to the surface, you can measure those changes in conductance in the sensor. So then the question becomes, okay, how do we make this graphene FET specific to our target biomolecule? And that's where we come up with this, or we use this P-based linker. So in essence, this linker molecule is, as I've mentioned, it links the aptamer to our graphene surface. It's just a pyrene group here on the left side that can interact with the pi bonds on the graphene surface, and you get basically pi-pi stacking interactions where they'll share electrons to the pi bond and create a relatively strong bond that way. Then you'll also have this free group here, this NHS ester group on the end, which will interact with a free amine group on the aptamer side. So if we change these aptamers such that at the 3' end of this single strand DNA end to have an amine group, a free amine group, we can create a peptide bond between this NHS ester group and that amine group that'll stick the aptamer to the surface of our graphene. And this way, we can create some of the specificity for the graphene through the specificity and sensitivity of our aptamer functionalized on the surface of our GFET. Once we have this sort of configuration where this graphene is modulated with a biomolecule specific aptamer, we need to transduce that signal and process it in such a way that we can measure out how much of these aptamers are bound to a biomolecule we're trying to detect. We can do this in a number of ways. One particular way is that we'll sweep the gate voltage across a particular feature of graphene known as the charge neutrality point. So as we change the gate voltage, we can basically create a plot of the current from drain to source that will look something similar to this. What you'll find in graphene is there'll be this charge neutrality point where the conductance changes dramatically at this point, and we can measure this point at a location at different concentrations of protein presence. And this way correlate the shift in this charge neutrality point to the concentration of our biomolecule being bound to our aptamer. A lot of the work I've done in this lab has been using a single GFET design. It's currently been put out to a company, Ampera, and this is the basic design of the GFET device. So we'll have a large gate terminal here, and then a source and drain across our GFET. This is a design where the gate terminal is actually, the gate electrode is in plane with our graphene channel. So these both will be exposed to the fluid itself. And the gate, as we sweep that gate voltage, will modulate the field effect across our graphene channel, and we can measure the change in that charge neutrality point or that Dirac point as we measure, take different measurements. So this is kind of an example of one of the applications our lab has done. We've taken aptamers specific to the receptor binding domain of spike protein, and another aptamer specific to the nucleocapsid protein of the SARS-CoV-2 virus. We took those aptamers and we adhered them to the surface of our graphene using that linker molecule. And then we were able to detect that shift in this Dirac point between the unbound state and the bound state of the spike protein or the nucleocapsid protein to our aptamer. These are some general results we came across. So there's a MERS-CoV, SARS-CoV, and SARS-CoV-2, just different coronavirus strains controls based upon the N aptamer and S aptamer binding. And these are all the Dirac shifts. So this is a measure in the change in the Dirac voltage between a baseline and a sample experiment. On the middle graph is just basic controls where we take a chip that has no aptamer on the surface of it, a chip that has basically a scrambled aptamer sequence, but same number of nucleotides, and also another that has only saliva in it, but still has the aptamer itself. And then we'll compare that with an S aptamer and N aptamer controls as well, and show that there's significant increase in signal between those different controls. On the right is a dose response curve where we'll look at different concentrations of our target protein, and we can use this to back calculate the limit of detection of our device, our biosensor. We've done similar experiments where we've looked at amyloid beta and tau protein and alpha-synuclein protein. These are different proteins related to neurodegenerative disease development and progression. Again, with different aptamers for each of these different proteins. We show here there are several different cross-reactivity controls we ran, where we take the amyloid beta aptamer, we cross it with the tau and alpha-synuclein protein and show there's a reduced signal compared to the proper protein aptamer pairs. On the right, we have, again, more limited detection and quantification results. Here, we showed that there's a limit of detection down to around 10 femtomolar in amyloid beta, significantly reduced number in the tau aptamer protein pair, but kind of a similar result with the alpha-synuclein to amyloid beta and around 10 femtomolar limit of detection. Limit of detection being the lowest limit we can detect the presence of a protein with a biosensor. So the question we've had after some of these single GFET experiments is we run across a few limitations in the single GFET kind of schematic. Biggest one is that it's small sample size. We have to run many tests to validate the detectability of a given aptamer protein pair. In addition, there's a lot of graphene and chip variability between different experiments, and this also reduces the number of samples we can go through for a given given experimental process. So then one thing we've come up with is developing a multiplex GFET array that will kind of reduce some of these limitations in a single GFET experiment. The first being that now we can, if we have multiple GFETs measured in a single experiment, we can use the averaging of those signals together to reduce the channel-to-channel variation we're seeing in a given GFET biosensor experiment. Additionally, this is probably even more impactful. We can independently functionalize sets of GFET channels to detect different analytes. So we could say, for example, have a certain, I guess we'll get to that later. So yeah, so we have proposed a CMOS IC integration. This is in the works. We're developing and designing this circuitry right now. The idea being that if we take a CMOS IC or a CMOS integrated circuit, take that sort of information and put it directly in contact with the GFET channels we're trying to measure across, we can speed up, average those GFET channels together, increase signal to noise ratio, reduce time to averaging as well. In addition, we can also functionalize or apply different aptamers at different locations on the surface of this GFET array. And this will allow us to detect different diseases or critical diseases with higher fidelity diagnoses. And it paves the way for maybe implantable technology in the future for biomarker monitoring, using maybe a plethora of different cancer biomarkers on a single implantable device for tracking cancer progression in patients at risk. One way we've looked into developing this GFET array is using a NISOC microcontroller. So this integrated circuit is called the Neural Interface System on Chip. It's used for neural stimulation of electrodes, but we're repurposing it for measuring and stimulating different channels of our GFET array. It's 32 channels, so we can basically run 32 independent channels across our GFET array surface. This is a comparison of the results between the previous design single GFET model and our current in development NISOC GFET array. We see comparable shifts across some controls here. The biggest difference being that for the NISOC, we've chosen a voltage or current clamp design rather than a voltage clamp. So we'll be measuring the change in voltage versus the change in current in our new proposed GFET array design. This is kind of the design we're working with right now, where we'll have a crossbar GFET signal schematic. So the biggest thing to take away from this is that we have the source and drain crossbars going perpendicular to each other in our design of the chip. So we can apply a source to drain conductance across a single drain crossbar and a single source vertical crossbar. This will allow conductance only across a single GFET channel at a time, whereas we keep the rest basically floating. So conductance, these won't be conductive in that state. Then we can basically iterate through each of these different GFET channels and measure conductance across each of these independent GFET experiments using this design. We went with this crossbar design mostly because we're limited by the 32 channels of our NISOC chip. If we wanted to design a chip with more channels, it would require more pads, and that requires a lot of fabrication process. So this crossbar allows us to basically do 16 squared different GFETs on a single GFET array with reducing the channel number. This is a current design for kind of a prototype chip that goes away from using, this was before we went to the crossbar design, where we have a drain connected to a common drain across all these GFET channels. Each of these GFETs has their own source channel connected to its own pad for wire bonding later. But again, like they're all common drain configuration, so this isn't a crossbar. But what this allows us to do is we're able to functionalize each of these different quadrants with a different aptamer, and thus we can detect 4 different aptamers with some repeatability, where there are 4 different GFET channels in each quadrant to measure independent signal across each of those. This is just to show some of the graphene transfer process. So it's a wet transfer process where we'll take graphene, we'll suspend it in water, transfer it underneath the chip we're trying to transfer it onto, There's a few annealing steps afterwards, and then we'll measure the graphene thickness using Raman spec. So we'll look at a peak here around 2700, compare it to a peak at 2600, this 2D to G prime peak ratio. Will basically tell us how thick the graphene layer is. The higher this ratio, the higher this 2D peak over here is compared to the 1600 peak. The more single layer the graphene is, if that makes sense. The less folds there are in the graphene, and the more monolayer it is. So again, this is pre-annealing step, so it's quite impure in some cases. Our crossbar design is this sort of configuration. So I mentioned we have the vertical crossbars. We'll have a dielectric layer with vias. So this dielectric is an insulating layer we'll put on top of the vertical crossbars. There'll be vias made of gold that'll connect the vertical crossbars to the top electrodes. These top electrodes will contain all of the pads for wire bonding our chip to its chip carrier. It contains the gate electrode. It contains the horizontal crossbars in addition to the pads for the graphene channel as well. On top of this, we'll put a passivation layer of SiO2. So we'll apply SiO2 uniformly, etch away the pads that we want exposed. On top of this, we'll then place the graphene itself, again, through the wet transfer process and then patterning later. On top of this will be a final microfluidic well arrangement where we can independently functionalize each of these different rows of graphene channels. So there'll be 5 different aptamers we can functionalize on a single chip at the end of this process. This is the crossbar fabrication cross-section kind of image, same process again, as I mentioned before, but this is a cross-sectional view of it. And this is the interface board we use between the NISOC and GFET array. So the NISOC, again, that's the microcontroller we're using to operate each of these channels, the different crossbar channels, and measure the conductance across those channels and also to modulate the gate electrodes. It sits within this chip carrier in the center here. It then breaks out the channels into this PCB, which then connects into this 36-pin edge connector on the side. We then wire bond our GFET array chip into the center of this chip here, and it can basically just slide into this 36-pin edge connector. So we can, once it's wire bonded on this chip, we can test at different times, different phases, and reuse the chip over and over. So this is more of a future design, but we're working on a design for the CMOS circuit. So we're developing a CMOS integrated circuit chip that can allow for independent gate voltage sweep. It'll basically measure automatically different graphene channels, GFET channels. It'll be, in this case, 16 by 16 GFET array with independently functionalized different GFET channels. This is basically a basic diagram of how the graphene deposition would work. We'd get the CMOS analog chip back from from fabrication. We could do graphene wet transfer, pattern photolithography at the end, and then quality control using Raman spec and AFM. And then the next step would be the functionalization with ssDNA aptamers. Depending on the size of the CMOS IC chip we get back, we have kind of 2 proposed methods we're going to look into. The first is more tried and true, which is a microfluidic well/array or microfluidic channel arrangement, where we basically physically put this well on top of the different channels, and then we can flow aptamer concentrations to different parts of the GFET array chip. The other is a photoresist method where we consecutively remove photoresist and apply aptamer in a consecutive sequential manner. And in this way, we'd end up with a selective functionalization process on the top of our GFET array chip. Then we do a few different validation methods, SEM-EDX, and maybe a fluorescent aptamer experiments to confirm localization of different DNA aptamers. And yeah, that's all I have to show today. Just acknowledgements for Gert Kauenberg's and Ratnesh Lau's labs, and also for the Fellowships Interface graduate training program. And any questions?
No questions?